Okay, we'll start now. Okay, ladies and gentlemen. Yeah, welcome to the Office of Information Management and Analysis monthly brown bag series. The webinar will be strictly 30 minutes event for both presentation and questions and answer. The title of today's webinar is the California Environmental DNA Program, Give a to Gate and Ecosystems. The presenter is Dr. Rachel Mayer, Executive Director of UC Conservation Genomics Consortium at UCLA. Rachel received her PhD in plant science from the University of New York and New York Botanical Garden, studying crop domestication. Then was National Science Foundation Plant Genome Postdoctoral Fellow at New York University. A AAAS Science Policy Fellowship landed her in DC working on the Dimensions of Biodiversity Program, National Science Foundation, where she realized eDNA had the potential to address gaps in ecology and evolution and even unite them. She joined UCLA in late 2016 to direct the UC Conservation Genomics Consortium, and through this runs the Cal eDNA program, does research on oak trees, ancient California, and helping to launch the Earth Biogenome Project for California. With this brief introduction, I'll pass the phone line for Rachel to start her presentation. Thank you, Rachel, for agreeing to do this webinar. So you can go ahead and...
Thank you, Dawit and Candace, for hosting. I really appreciate being able to share some news about the Cali DNA program with all of you and to potentially start some collaborations from this. So I'm going to just jump in. Feel free to ask questions at any time. So we know that we are losing a lot of biodiversity in this century, and we need to do something about it. The University of California is involved in many different programs across the state and national and international for biodiversity monitoring and potentially management, and also the basic science to understand the fundamental questions of how does biodiversity, how is it maintained, what shapes it. The University of California recently launched a conservation genomics consortium that's a little bit more applied, trying to develop genomics tools to actually be easily translatable and adoptable to solve some of the more management-oriented questions. And then also the Earth Biogenome Project launched with one of the founders, Harris Lewin at UC Davis, which is an initiative to sequence high-quality genomes for every family of eukaryotes on the planet. So we realized that with all of these great sequencing initiatives, and many of them happening in California, that there is going to be a lot of data for the rich species that we have here, where we could actually use the DNA data to perhaps scale up our environmental observations. So we know that satellite data, airborne LiDAR data have been tremendously powerful to be able to predict environmental change and also to find hotspots of biodiversity that could be conserved that maybe hadn't been identified before. Environmental DNA might present an additional layer. Right now, we don't have a way to real-time monitor biodiversity at any sort of scale like abiotic data. But what if we could involve the public and actually really have big data where you could understand migration patterns or what's in your neighborhood, sort of like you check the weather every day. iNaturalist and other citizen science platforms have been incredibly helpful to leverage the energy that the public has to help their environment become more resilient, to turn that into data. So there are over 1 million people that use iNaturalist. Many of them are in California because it launched in California. It's managed by the California Academy of Sciences. And it's very simple. Everyone has a smartphone, so you can take a photograph of an organism, connect it, share it online with other fellow naturalists who could be informal, like hobbyists, all the way to the taxonomic expert for that millipede that you photographed, waiting for someone to put a photograph of an insect online or some other bug. And then you have a platform to discuss your findings. So this not only engages people in data collection, but also in the analysis. And so now we have this community of people that are really primed to be able to do more, to be able to build a strong feedback loop with professional scientists and monitor the biodiversity in their local areas really well. We also have the California Naturalist Certification Program that's got, I think, over 40 different campuses in the different biomes of California where you can get local naturalist skills. So we have this incredible workforce here. What we want to do is help people understand what's here, what defines the habitat that they're in, and to be able to track if that habitat is changing. And we're really excited that we don't need to go monitor the mammals and the insects and the spiders and everything separately. We can just look at dirt, because in soil or in sediment or in water is loose environmental DNA from all of the organisms that were physically in that dirt sample, like maybe some of the bacteria and archaea and fungi, as well as organisms that shed DNA that may have passed through. Environmental DNA has been really a diverse technique. It's been applied to social justice issues, to understanding geochemistry and ecosystem cycles. It's been very important for understanding the microbiome for human health and animal health. It's really got a lot of potential to connect different disciplines of science. And we're really excited because to collect it, you're just putting gloves on, filling a tube of soil, or filling a bottle of water. And so it can be really easily crowdsourced, and it can be used to go back in time. So I actually have a project that's not related to CaliDNA, but going through sediment from lakes in Southern California from the Pleistocene to the current day. So we can go back like 34,000 years with environmental DNA if it's in conditions where it's preserved. But on the surface, we have a nice record of, say, the last 6 months to a year of what's been in that location. So it can be a very local signal, which can tell you a lot about the local community in recent time. So the CaliDNA program is based on that anybody can volunteer as a community scientist. We use the term community scientist because citizen scientist isn't really in vogue anymore because nobody wants to accidentally imply citizenship to volunteering. People can come to us and say, I want to get trained. I want to learn how to collect soil samples or sediment samples. And so there's lots of training that we offer online, and we also do training at BioBlitzes, which are group events where everyone goes and hikes around and takes iNaturalist photographs and also collects samples. And they're a lot of fun. We also have a big undergraduate internship program where we teach people how to do all of the laboratory work and analysis of eDNA. And we're working with other organizations like SWRCB and people at the Natural History Museum, at Cal State campuses, to also try to kind of unify our approach to the environmental DNA informatics pipeline so that we can make data more interoperable. We're doing DNA metabarcoding, but like I said, like I mentioned with the Earth Biogenome Project, if there are eventually whole genomes of California native species that we want to monitor, we can leverage those entire genome resources with the DNA that we've extracted and not only look at species, but look at functional genes and look at demography. So we might be able to even get population level information from the DNA. So right now we're just doing metabarcoding, but there's a lot of opportunity to move environmental DNA into really cool uncharted territory, starting in California, because we have these initiatives launching here, and so many scientists to focus on the biodiversity that we have. So again, what I'm calling the parabiome is right now a metabarcoding approach that uses 5 different markers. 16S, which is frequently used for bacteria and archaea. 18S, targeting broad eukaryotes. It's great because it targets broad groups, but it doesn't have very high species level resolution. ITS1, which is targeting mainly fungus. And then the CO1 mitochondrial region, which targets animals, but also gets oomycetes very well, which includes things like Phytophthora. And then the second ITS2, which targets angiosperms, but also we get gymnosperms really well. We don't get ferns very well with that. And the MyFish 12S primers for fish. We have some additional primers that we occasionally use if we're working, say, in the desert where we have higher rates of degradation, like TRNL. So we have the options to also look for organisms using shorter DNA fragments that you can pick up in a more degraded sample that may have been exposed to harsher elements. A lot of the team from the UC Conservation Genomics Consortium has really helped advance the precision of our lab work. So for example, the different polymerases that you could use when you're doing PCR, they can introduce a lot of bias depending on the GC content of your DNA templates in your sample. And so this is a big problem if you're especially looking at relative abundance. So on the right, you'll see some numbers on the yellow, in the yellow. Those are the numbers of papers that use the polymerases in the graph. And you'll see that 381 is for the fusion polymerase. That has a very low R-squared value, meaning it has a lot of GC bias. Your observed and expected proportions are not good. We use the Qiagen hot star polymerase, which almost has 0 GC bias, but it's not very frequently used yet. So we're trying to make recommendations based on these findings so that people can all kind of come together to use some of the best products, and we can also help relay that to the companies that make them. Here's an example of that. Oh, I'm getting feedback. Okay, sorry. Of what can happen if you have GC bias. So each line, these are qPCR graphs. Each line is a different plant DNA template and how much it amplifies with every cycle of PCR. And so what you start with at 20 cycles ends up looking very different in terms of relative abundance to 40 cycles because of GC bias. So we try to reduce that. And that work was mainly done at UC Santa Cruz with Beth Shapiro. Then we also are trying to build tools to help the public, from natural reserve managers to students to everyone, to really engage with the data and not just look for their favorite species, but try to understand that it's all about the patterns that we see. And we really need to look very broadly at sort of a system level. So we have this website, ucedna.com. Please go to it. We're still improving it all the time. We have a great programmer that works with us. You can explore the samples that have already been collected and see the biodiversity in every single sample. So if you're a volunteer, you can track your sample, and eventually when we process it, you can see not only the photograph that you took, but all of the taxa that we matched to the DNA sequences. We get on average about 1,000 unique taxa per DNA sample. What we're also building are tools to relate environmental DNA to other types of observations. For example, the Global Biodiversity Information Facility, or GBIF, houses the iNaturalist and eBird observation data. So how do we fit in with all of the other community science data? that's being collected. So on the top, this is a case study I'll be presenting to you in a few moments. This is Pillar Point at Half Moon Bay near San Mateo. And we have 88 eDNA samples that were collected by volunteers. This was our first BioBlitz in collaboration with Cal Academy of Sciences and the Natural History Museum of Los Angeles to sort of look at how iNaturalist observations and eDNA observations relate to each other. So on the bottom are the iNaturalist observations, and you can see that they are much richer than the eDNA observations. And on the left, this left polygon, the red highlighted polygon that's drawn, that's a marine protected area. And so humans are not supposed to just, from the public, are not supposed to just wander in there, although people do a little bit, but it has far fewer observations, photographic observations made with iNaturalist than, say, this blue polygon, this blue polygon where there's unprotected tide pools that school groups can go to. Everyone can sort of go out in low tide and really look at what's in the tide pools and explore. So it's very heavily frequented. Every month, iNaturalist BioBlitzes occur there. And then on the right is this embayment where you have a marsh where there's a lot of ag runoff. And you also have a lot of boats that are docked. So you have actually quite high bird observation diversity here. So how do these photographic observations relate to eDNA observations? Another thing that we're doing and we're trying to build out is when we see environmental DNA patterns. For example, we have a transect that was coastal, shrub, scrub, and forest, lowland forest in California. We found, for example, from a random forest analysis that the amount of silt or elevation are different factors that are highly predictive of the environmental DNA signatures that we find. And we can explain how much each family across all of the tree of life is predictable by these abiotic factors. So how do we get the public excited about those results? Well, one of the things that we have on our website, and this will become more powerful as we scale up, and eventually we can make models from this, say, species site occupancy models, or species distribution models, is you have different map functions. So my recent favorite thing is diatoms, so we'll look at those. So Bacillariaceae, these you can look on our Explore data page, type in the name of a taxon that you're interested in at any level of classification, and you can see out of the samples that we've sequenced so far where it's been found. And then, okay, so maybe not everyone cares a lot about diatoms. The group I'm talking to right now probably cares very much about diatoms. I recognize that. But we'll move to tardigrades as an example. So here we found tardigrades in 58 samples, and this is something like every student wants to know, how do you predict where tardigrades live in California? Because everyone wants to find them, even though if you found them, you wouldn't be able to really see them with your naked eye. So we still don't know, but this is something that as we accumulate data, right now we have 58 samples out of the 500-something that have been sequenced and put online where tardigrades have been found. And so eventually we'll be able to find the environmental predictors for tardigrades. And those are projects that we want to hand over to students or to the public to do, and then to have a feedback loop with us so that we can also guide more BioBlitzes in that direction and really figure this out. So we have different maps that you can go through to start to generate your own hypotheses. Oh, is there something about human impact that relates to tardigrades, or are they sort of less frequently on the coast and more inland? So just to refresh, CaliDNA, you do some training. We send you a kit that has gloves, tubes, a straw, and then you use your app to collect the metadata while you're collecting your sample. Everything's pre-barcoded, and so then you just mail it back. We give you some meters where you can collect pH, temperature, other data, and then you just take photographs. So it's a pretty easy, like, low bar. to complete a kit successfully, and then we pay for all of the shipping. We've had lots of volunteers, about 1,000 volunteers now. We also have a software that is being used by about a dozen graduate students and people at different institutions called Anacapa, named for Mirage Island, which is what Anacapa means in Chumash. And that's because maybe eDNA gives you a signal that you don't know how accurate it is. So we had some jest with that. And then we also have some online community ecology statistical analysis tools like Ranacapa, so that's R, Anacapa. That's a Shiny app, so we can actually give people the data sets and let them do alpha and beta diversity analysis. And even if they have no background in community ecology, it's pretty intuitive to interpret bar plots and ordination plots. And we found that managers, especially UC reserve managers, where we've done a lot of these bioblitzes, they really want to engage with the data to see how their reserve compares to other nearby UC reserves. We've also started a collaboration with NASA JPL climate scientists and engineers to use their hyperspectral data that they've been collecting for different parts of California and see how that helps improve biodiversity predictions with eDNA. On the bottom right, you'll see an image of a burn zone. So we actually have a Woolsey Fire project right now that we're hopefully going to get some extra funding for, to be able to track biodiversity as it comes back and to understand the role of refugia in feeding the biodiversity in the burned surrounding areas. So I have about 9 minutes left. I'm going to jump into a case study, that Pillar Point case study. And before I do that, I just want to open up for any questions right now before I continue.
I have a question. Can eDNA detect species that have not been discovered yet?
Yes.
And is that confusing, or how do you deal with that?
Yes, that's a great question. So I'm going to jump a couple slides ahead, and so right now, environmental DNA, what we do with it is we match it to all known publicly available sequences. And so we are only as good as our reference database. So we might be able to say with high confidence something is in this clade, maybe something looks like it's inside a certain genus or family, but we can't match it to any species. So that, what we're trying to do is create a communication loop where then we tell people that are making voucher specimens and doing DNA barcoding, hey, we found a sequence that doesn't have a match. It's probably a nematode. Can your nematode specialist go collect this sample, look at this environment and see if there are new species there? But also we have a good understanding of the gaps across the phylogeny of where we really don't have very good sampling. Another thing we can look at is genetic diversity aside from taxonomic diversity. I didn't put a slide in here, but you can see, for example, under ostracots that there sometimes is much more genetic diversity than we can assign. So there is a relationship between taxonomic diversity, even if we make an assignment that is like actually a sister species and not the actual species. We can say, hey, there's a lot of genetic diversity that maps to ostracots but not to family or genus. This might be something really exciting, and then we can point people to that to look. So right now, all metabarcoding analysis software starts with the leaves of the tree and works then back to towards the root. And that's actually the inverse of what you want, but it's the most computationally easy. But Rasmus Nielsen and his postdoc Lenore Pipes at UC Berkeley are part of the consortium, and they've built a software that starts at the root of the tree and works to the tip. So then you actually don't have a best hit match that is actually very imperfect. So one of your problems is you might have your query sequence and you can't tell if it's in a clade or next to a clade. So this reverse approach, it's finally computationally possible, and they've made it very fast, and they're going to be publishing it soon. So we're really excited to share that with people. Does that help at all?
Yeah. Thank you.
Okay, great. Thanks. So this study on Pillar Point, there's a link here. It's not something that we've shared widely with the public. We're still building out tools, but this is like our big beta testing site where we're trying to see how photographic observations by the community scientists and eDNA observations by community scientists can really complement each other. And so we are using GBIF that I introduced earlier, and we've also used some very special DNA barcode sequences from coastal invertebrates that the Natural History Museum of Los Angeles has collected. So these were DNA barcodes from California, and we wanted to see how much they also enriched the results that we got. So I can't go into too much detail, but what we do see is the diversity profiles of the different sections of Pillar Point, that protected area, that unprotected area, and the embankment are really very different looking from each other through the lens of eDNA and GBIF as you go through taxonomic ranks. And that's because GBIF is mainly observations of animals. As a botanist, I always say people are plant blind, and this is totally the case for photographic observations, is we don't have very many people reporting, inventorying fungi, plants, algae, and other photographable things. But for eDNA, and of course for eDNA, maybe we're not as good at recording, inventorying, say, the birds, but we have a good record of the bird mites. So we're getting smaller organisms. We actually get more of the lower trophic levels. So in a way, we really can complement our results with the photographic observations of bigger, more mobile things that aren't really ubiquitous in the environment. And so here's a graph just presenting some summary statistics of how we align. So eDNA, we tracked 82 phyla. GBIF tracked 19 phyla. Of the GBIF records, all 19 phyla were in eDNA results as well. We had high concordance at the level of class, the level of order, and then once you get to the level of family, you start to see a split. And one of these questions was, is that because we're truly detecting different things, or is it because we're limited with the sequences that are available because there's some bias? Well, it turns out that the bias is very much the same. Also, we have to recognize that photographic observations are often being matched to species by machine learning algorithms or deep learning algorithms that use databases that are also very limited in predicting what that species is. So we're all just working with like the best shot that we can, but we have limitations, genetic references that are limited, or maybe morphological characters that we're limited in looking at from a photograph. So here on this plot, you can see that we don't really have these singleton observation tails in phylum, class, or order, but we do as we get to genus and species in both GBIF and eDNA. I think it's 12:30 now, correct?
Yeah. But
I have 2 minutes.
5 more minutes, yeah, with the question.
Okay.
Yeah.
Okay, great. So this is my last slide. One thing that we've shown is that we can find in places where we don't have very many photographic observations, like the protected area of Pillar Point, we can find that it's highly similar based on eDNA to the tide pools. So what's nice is we can say, okay, if we can do observations here in the tide pools, we can actually predict that they look very similar to the protected area. And then we can also pull apart the species that are in the protected area that are not in the tide pools and highlight what's there. With network analyses, it's been really incredible to find these functional associations that help us understand that actually the tide pools, the protected area are functioning very differently. And in the polluted embankment, of Pillar Point, we get haphazard biodiversity, where you see many more different colors in this bar plot, where sort of you don't have a regulated system anymore. So we have beta diversity analyses as well, where we can show that abiotic factors really are good predictors for a lot of different family level and class level designations. And I'm going to leave it at that. Thank you so much, and I'll take questions.
Yeah, we have some time for questions. If you have questions, you can jump in.
Or you can type it in the text box.
Is there any question there?
No.
So right now we're just exploring lots of different analyses. We're trying to publish some papers. We're trying to scale up. We have undergraduate education modules that we've built. If there are bio-blitzes that you're interested in running or participating in, we'd really love to connect, and especially to connect other communities in California that are doing environmental DNA.
That's good to know. And if there is no...
I have one question.
You have one question.
Yeah,
Great.
I do.
Thank you.
So if you see different biodiversity among organisms in protected environments and adjacent unprotected environments, are you able yet to draw a correlation between what the characteristics of the protected environment that are allowing the more stable display?
Yes, I think we are getting there. For example, the network analysis showed that the higher amount of kelp in the protected environment was sustaining a specific community of ammonium oxidizing microbes, and so those were in turn sustaining other organisms.
Mm-hmm. Okay,
so we are starting to see that.
So I hope papers on that.
Thanks. Yeah, we've seen more in the protected area. We've seen more anemones, more other echinoderms, and I'm going to, gosh, I can't ever remember the Latin name for the life of me of this one group. They're like basal chordates that look like little polyps, but they're actually like vertebrates. They're beautiful. I'm just basing on the name. Pardon?
Tunicates?
Yes, tunicates is in the group, but it's yes.
Okay.
So we see more of those in the marine protected area as well.
Yeah. There is one more question.
Sure.
Okay, this is from the chat. What would be the temporal resolutions of eDNA for different taxa? Could months or seasons or time of day matter?
Well, that's a great question. For eDNA, as soon as DNA is outside of a cell, it will start to break down as soon as you have water, light, UV, heat affecting it, or sometimes other microbes that eat DNA for breakfast. So we definitely have seen that you have a shorter temporal sort of decay rate in the desert versus say the forest, but once that DNA is loose, it shouldn't decay differently for a mammal versus a plant versus a microbe. So I think what we're seeing is, even though the temporal length may vary a little bit, and it's actually quite predictable by precipitation and other abiotic factors, we do think that we're seeing a scale where all the organisms that we're inventorying are from at least the same time window. So we're trying to ask questions that are appropriate to the design of, or just to what eDNA is, and really asking things about community, and not necessarily about when something was present, until we have enough data where we can actually tease that apart. And that is a goal. I think when we get, a couple thousand samples, we'll have a really clear picture of what's driving the decay of eDNA.
Great, thank you. I think that's all the time we have.
Thank you. I really appreciate this opportunity.
Sure.
And if you have any following questions, just send me an email.
Okay.
Okay, thank you very much.
Thank you very much, Rachel.
Thank you all. Have a good rest of your lunch. Bye.
You as well.
Bye.